So far we talked about how cells that are right beside each other, adjacent to each other, communicate. We call that local signaling. And if we were talking about animal cells, one way they communicate is through the junction between the two cells, the intercellular junction between them. And what's that called in an animal cell? The junction that lets them communicate. They're side by side. It's called a gap junction. And I said a good example, just to know of that, if you had to like write an essay or something, would be what cell? Heart. Cells in the heart. That way they can sync up what? Sync up their beating. They can beat in rhythm. A very similar thing to a gap junction in plants is a connection between adjacent cells called plasmodesmata. Think of these sort of like tunnels that go through the walls and connect up the cytoplazms. And they can transfer materials or signals, messages, through those little tunnels. Another thing we spent a lot of time talking about was how the immune system does this local signaling business. And I want to spend some time talking about that again now. This purple cell up here, what kind of cell are we looking at? A macrophage, which is a white blood cell. And it's a white blood cell that phagocytizes or eats foreign things. What's the name for any foreign thing in your body? Antigen. And what it's going to do is this cell is going to eat that virus or that bacteria, put it in a vesicle, digest it using enzymes from the lysosome, intracellular digestion. And normally, when the lysosome does that, what happens to the pieces of most things that cells digest? You either use them to make energy or you use them to build cell parts. But in this case, what happens to at least some of the pieces of that virus? They get held outside. They get put up on this green protein, which is not really green inside the cell, but that thing is called what? MHC class 2. And the job of that protein is to put those viral or bacterial antigens on the outside to signal certain things. What's the process of taking those antigens and putting them on the outside called? What's the process where the virus takes the piece of the virus and holds it up out there? What's that called? Antigen presentation. So the MHC class 2 is a piece of a macrophage that carries out this process of antigen presentation. Who's it presenting the antigens to? Helper T cells mostly, and that's what we see docked up. This down here would be a helper T. And what's the helper T learning during this process of antigen presentation? What the virus is like. It's learning its shape, its charge, different things about it. And once they undock, once they unconnect, what's the helper T cell going to go do? Yeah, it's going to go spread the message. We sort of think of it like the Paul Revere of the immune system. It's going to turn on other parts of the immune system. What disease wipes out your body's helper T cells? That's what HIV does. And without these helper T cells, pretty much none of your immune system works. That's why AIDS is so serious. Now, only cells like macrophages have class 2 MHCs. Their own specific white blood cells. But every cell in your body has a very similar protein, like this one, called an MHC what? Class 1. You might think of these proteins sort of like the cell giving itself a physical. Your cells are constantly taking proteins from inside themselves and displaying parts of those proteins on this MHC class 1. And who's coming around checking those proteins? Helper T's and killer T's. We're checking them out. If it's a normal, healthy cell, what kind of proteins are being displayed? Normal, healthy proteins. So the killer T says, hey, you're looking good today, have a nice day. But on the other hand, let's say that cell up there is infected. It's got a virus inside. What are some of those proteins that are being displayed probably going to be? Virus proteins. And very quickly, your body's going to recognize that. And the killer or cytotoxic T cells are going to do what to that cell? They're going to slice it up. They're going to kill it so that what doesn't get finished? The What cycle? The lytic cycle. Because if they let that cell live and let the lytic cycle finish, viruses are going to burst out and kill other cells. They don't want that to happen.
But wouldn't they slice the cell up and maybe wouldn't outwish the viruses?
Well, two things. One, the hope is they slice it up before the process is finished, which means the viruses aren't working. But even if some of them are working, what can get to the viruses outside the cell that can't get to the viruses inside the cell? What's your body's main defense? What do you make against viruses? Antibodies. Remember, they're big old proteins. They can't get in the cell, but they can work outside. So we've at least cut it open so the antibodies can get in. Right. So again, the immune system's kind of complicated. There's two main parts of what's called your specific immune response. The antibody-mediated part. Camera, let's focus back there, please, sir. And the cell-mediated part. You can't make it very far without either one. Let's say that you've got a virus infection. You've got antigens in your body, foreign proteins. It's the first time you've ever had that particular virus. That germ gets eaten by macrophages. They digest it, display their antigens, present the antigens, dock up with that helper T cell. The helper T cell kind of goes Helen Keller on that antigen, figures out what's going on. And then notice that helper T cell is critical because it stimulates both parts of your specific immune response. Neither side of this diagram will work without these helper T cells. The side on the left, the helper T cell activates B cells, which eventually make antibodies. Antibodies that are specific to what? That particular, what's the word? Antigen. They only attach to that thing. And when they attach, they help in some way or another to kill or get rid of that germ. But they can only get to where? Outside cells, because they're big old proteins. A word that you need to recognize, this word shows up a lot in this class. I don't think we've seen it yet. Interstitial fluid. What do we know inter means? Between. This is the fluid between your cells. This is where antibodies work. In the interstitial fluids in the blood, in the lymph. But in the fluids between the cells. Interstitial fluid. Also notice though over here on the right, the helper T cells stimulate these cytotoxic or killer T cells. What do the killer T cells kill? Your cells that are what? That are infected, so that they don't spread. So without both of these parts working correctly, you're probably never going to get rid of a virus. You need both parts working together. When the infection's over, both parts leave behind these memory cells, which is huge. If it's the first time you've ever had an infection, how long does it typically take you to do all this and get rid of the infection? Yeah, maybe up to a week, maybe longer. Because your body has to figure out how to defeat that thing. It has to make the antibodies. It takes time. But once you had that infection, these memory cells are floating around. And if that same exact germ gets in you again, what happens? You kill it almost immediately, so fast that you don't even get any symptoms the next time around. Pretty cool. So no memory cells are there. They protect you the second, the third, the fourth, the fifth time you get that same infection. Something else that we talked a lot about were nerves. What's the name for a nerve cell? Neuron. And neurons are typically transmitting what kind of signals? Electrical, right? Electricity. Same electricity that runs this projector. Same thing. The neuron has three main parts. The cell body, or soma. These parts that look like little antennae, which essentially is what they are. They're dendrites. And then this piece that comes out, the single piece that comes out, and that's called the axon. Now, in general, the dendrites are there to receive signals. The signal is going to tend to flow this way, from dendrite toward axon. What's a dendrite going to be connected to? No. Not usually a muscle. Remember, it's receiving signals. It could be another nerve, or maybe it could be nerves coming from your eye. Maybe it's receiving visual signals. Maybe it's nerves coming from your ear, from your nose, from your taste buds, or maybe from some kind of receptor in your finger. Regardless, it's receiving information. That information is transmitted as electricity down this axon. And the end of the axon is going to be connected to what? Another nerve, or maybe to a muscle. But is it directly connected like that? No. What's the connection called? A synapse. And it's actually like a little gap, right? Looks like this. So this would be the end of one axon. And this would be the beginning of the dendrite that it's attaching to. And there's that little gap in there. And that little gap's the synapse. Remember the way this works. Electricity runs down this axon. It gets near the end, and that stimulates this neuron to release what into that gap? Again? Neurotransmitters, chemical signals, ligands. And those neurotransmitters will diffuse across that gap and bind to what on the postsynaptic neurons? Receptors. And if enough of them bind there, what happens in that next nerve? What starts in that next nerve? The signal. The electricity starts. And we're about to figure out how that happens in just a minute. But the key thing, electricity in the presynaptic neuron causes it to release neurotransmitters. Those flow across the synapse, bind to receptors. If enough of them bind there, that causes that signal to be transmitted over to the next nerve, the next neuron. We also spent some time talking about long-distance signals like hormones and pheromones. And I need to step out for a minute. Take this. All right, let me try again. So, long-distance signals, things like hormones that travel through what? Through the blood to every cell, but only affect cells with the right receptors. This slide is the most important one. I've said that 100 times. I'll say it some more. Know the steps of cell signaling. First step is reception. What's the word for any kind of signal in a cell that can't get into the cell itself? A ligand. Okay? Ligand or ligand. The ligand binds to the receptor. Where's the receptor usually at? Outside. What's the word for a receptor that's outside? Extracellular. And they have to fit together kind of like what? Puzzle pieces, lock and key. And when they fit, what physically happens to that receptor? Changes shape. And what's the word for changing of the shape? What kind of change? A conformational change. Conformational change. And that triggers step 2 of this process, which is transduction. Step 2 is by far the most complicated part. It's a lots and lots of steps. It's a process. Sometimes it's called the signal transduction pathway because it's so multi-stepped. Key things to know right now is that 2 main things happen in transduction. What 2 things have I stressed again and again and again and again? Translation of the message and amplification of the message. Know those 2 things. We'll get more details in a second. The whole purpose of this signal, though, is to cause some kind of response. And the response can be lots of different things, but usually it's either turning on or turning off a gene, or maybe turning on or turning off a specific kind of like chemical reaction. So turn on or off a gene, or turn on or off a certain reaction. That's the typical response caused by a cell signal. The stuff on this slide I think we mostly talked about already. Remember that cells, signals on the other hand, only affect the appropriate cells. And how do we know they're appropriate? What makes them appropriate? They only have, those are the only ones with the right receptor. Remember that ligands are signals that are mostly going to bind to extracellular receptors, which means more than likely they're made out of what? What are most ligands made out of? Proteins. Too big, too charged to get in. Again, remember they bind with a receptor like a lock and a key. That causes that receptor to physically change shape. Usually the receptor will run all the way through the membrane. So there'll be a piece sticking out on the top, and it'll go through, and there's a piece sticking out the bottom. Where's the change in shape usually happen? The piece in the bottom that's inside the cell. So that actually transmits the message from outside to inside. So that's one of the key things that happens. That message is transmitted from outside the cell to inside. Remember, there are a few signals that have receptors on the inside of the cell. And those receptors are called intracellular. And what kinds of things are we talking about there that have? Say again? Specifically, which ones? Hormones. Specifically, steroid hormones like testosterone and estrogen. Because what's the membrane mostly made out of? Lipids, phospholipids in particular, right? What happens when a lipid touches another lipid? They go straight through each other. So the steroid goes straight through. What would be the advantage of having a receptor on the inside instead of on the outside? It's faster, and it doesn't have to go through all the steps that we've been describing. And it also can make a bigger response, a faster, bigger response. You go to a gym where you got all these muscle heads working out. Those guys might come in the size of Kool-Aid, 1 week, and then 2 months later, they're huge. That's not natural. That doesn't happen naturally. No matter how hard they're working out, that doesn't happen. Okay? They're juicing. Okay? They're taking steroids, and it happens. But it causes a fast response because those hormones go from the bloodstream straight into the cells. I'm going to skip that part for a minute. I'm going to try to focus on transduction here for a couple minutes. Remember, it's a many-step process. Kind of complicated. The benefit of the way this works, though, is just a few signals, a few ligands, at the end of transduction can cause multiple responses because, like we said, it's sort of like a domino chain. That you start with one domino, but that chain starts to split and split and split and split and split. And by the time all the dominoes have fallen, thousands and thousands may have fallen because of that one in the beginning. That's sort of the way this works. Kind of a good summary of what signal transduction is that one little statement right there. It's a series of steps in which one protein activates another until the desired response is triggered. Again, think of it like a chain of dominoes. Also remember, like it says up here, that most of the time that the signal, the ligand, never goes into the cell. It binds to a receptor on the outside. And that triggers all these things to happen on the inside. We're going to watch a video later if we have time. How many of you watch the Mythbusters? Anybody watch the Mythbusters? You guys seen those crazy, complicated machines they had built to do something really simple? You know what I'm talking about? You guys know what those machines are called? They're called Rube Goldberg machines. Transduction is sort of like a Rube Goldberg machine. There's all these crazy steps that have to take place, and then at the end, something like break down glycogen into glucose is what you're trying to make happen. But it's a complicated maze of things that have to happen to get that response in the end. A lot of times, these steps are turned on during the process by phosphorylation. And let's remind ourselves, what does phosphorylation mean? Phosphorylation means take ATP, which we're going to use these markers to represent, break off that final phosphate, give it to another protein, your protein. What did we just do to Dylan? We gave him energy, we gave him phosphate, we said, Dylan, time to go to work. They activated it. And in this analogy, what am I? Since I'm the one who gave it to you. Now, what are the enzymes in the cells called that actually give phosphates to things? They're called protein kinases. That's a key thing to know. Protein kinases are, since it ends in ASE, what are they? They're enzymes. Enzymes that take phosphate from ATP and give it to other proteins inside the cell. And thus turn them on, activate them, make them go to work. And the process of giving the phosphate to something is phosphorylation, right? Phosphorylation turns things on. Mainly because it gives them what? Energy. Phosphates are a way to transfer energy. Think of phosphate like the money of the cell. I'm paying Dylan to go to work right now when I'm giving this phosphate. Go to work, Dylan, here's your salary. That's what phosphate's like. It's like the money of the cell. All right, so this is kind of what we just talked about. That kinases are enzymes that transfer phosphate groups from ATP to a protein. And this usually helps to activate them, turn them on. Now these things are really, really important. Up to 2% of all your DNA codes for these things. Well, they're enzymes. Enzymes are made up of proteins, and proteins are coded for by DNA. These are really, really important enzymes. A lot of these kinases regulate things like cell growth, mitosis, meiosis, and cell reproduction. And if you get mutations in the genes for these kinases, what can happen? Either they don't work fast enough, or more likely, they work too fast. And what happens when you have protein kinases working too fast? It'll get cancer. It's one of the most common causes of cancer. Because cells start doing what too fast? Reproducing too fast. And that's what cancer is. So it's one of the most common causes of cancers are mutations in the genes for these enzymes, protein kinases. Yeah.
What are signs of cancer?
Well, I mean, it could happen anywhere in your body. But I mean, this is where you go to get tumors and such. The opposite of a protein kinase is a protein phosphatase. We might say that these two enzymes are antagonistic to each other. Which means they do what? They're opposites. They work against each other. Protein kinases give phosphates. Phosphatases take away. Protein kinases give energy. Phosphatases take away. Protein kinases turn on things. Phosphatases turn off things. Together, they help to regulate them. So it's a team. These turn on, these turn off. All right, now, like I said before, this process of transduction is pretty complicated. And it oftentimes happens in a series of complicated steps, almost like a Rube Goldberg machine. It's called the phosphorylation cascade. Because essentially, this protein right here is turning on this one, which then turns on this one, which then turns on this one, which then turns on this one. But this makes it look like it's a straight chain. This one's not just turning on this one, it's also turning on another one just like it over here, and another one just like it over here, and another one just like it over here. So it's like mini snowballs starting at the top of the hill and all rolling down. And it just gets bigger and bigger and bigger and bigger and bigger. Until at the end, eventually, hopefully, we get what? What are we trying to get? What's the purpose of the signal? What's the last step? A response. And hopefully, what kind of response are we going to get? Because of all this splitting that's going on. When I'm talking about the splitting, what are we trying to show? Amplification. So we should get what kind of response? A big one. Okay? A big response. Because it's branched and branched and branched. Let's see, that we're not going to worry about. Let's talk a little bit about what he talked about in the video that we watched in class yesterday. He was talking about what hormone in the video? Epinephrine, also known as adrenaline. Made where in your body? Where's epinephrine come from? What glands are right up in here? Your adrenal glands. They sit right on top of each kidney. Make adrenaline. That's what they call adrenal glands. And that adrenaline will flow through your blood, so it goes to every cell in your body. When do you release a lot of adrenaline? When you're scared, when you're in danger. And it's supposed to cause what response? What's adrenaline supposed to cause? Fight or flight. Fight or flight response, right? If adrenaline makes it to cells in your heart, what's the message it tells your heart? Okay. What are the tubes that lead down into your lungs called? Well, yeah, but the branches off of that. Called bronchioles. Bronchial tubes. What do you think adrenaline tells those tubes? Dilate. Get wider. What do both of those things accomplish? Widening your breathing tubes and speeding up your heart. More oxygen to your cells, and therefore more, back up a step, more what process? More cell respiration, and therefore more ATP, so you're ready to what? Fight or run. One of the two. So it comes down to energy. What if adrenaline made it to cells in the pupil of your eye? What's the message there? Dilate. So you can see, because a lot of times we get in trouble at night, right? So we need those pupils to dilate so we can see a little bit better at night. But in the video, he talked about cells where? The effect of adrenaline on cells, where at? What was that whole video about? Well, in the liver, okay? Cells in the liver. So let's say this is a liver cell we're looking at. Here comes adrenaline. Fits into that receptor, just like a lock and a key. That's called a G protein receptor. That's a specific kind of receptor adrenaline works with. Remember that once it binds, what does it cause to happen to that G protein receptor? It changes shape. And in particular, a piece of it breaks off, called the alpha subunit. And that goes across the cell membrane and gets to this enzyme called adenyl cyclase. Notice that this alpha subunit is carrying GTP, which is pretty much just like what? ATPs. Same thing, essentially. And what's it going to do to this adenyl cyclase when it gets there? And what's the word for that? What's GTP going to hand off to the adenyl cyclase? A phosphate group. This is going to get phosphorylated, which means now it has what? Now it has energy, and now it goes to work. And this particular enzyme takes ATP and converts it into this thing called the second messenger, CAMP, cyclical AMP. This diagram makes it look like it just makes one of them. It actually makes bunches of these CAMPs. So there's more what? More what? If we're making lots and lots of them, we're doing more of what? More amplification. These CAMPs are going to go and activate certain protein kinases. And those are going to activate more protein kinases. And those are going to activate more and more and more and more and more. Until eventually at the end of the chain, what gets turned on? What are we trying to turn on here? An enzyme that will do what? Break glycogen and release glucose. Release glucose to where? To the blood. And once that glucose is in the blood, what are you going to do with it? You're going to use it to do cell respiration, make ATP, so you'll have energy to fight or flight. All right, make sense? So transduction is a pretty complicated process, and it involves this whole cascade of phosphorylations where one protein turns on another, turns on another, turns on another. Pretty complicated. Couple of final points here. Usually when you hear the word secondary messenger, I want you to think of CAMP. CAMP. There are some places in your body, though, where calcium is also a secondary messenger. Calcium ions, Ca2 pluses. And those are particularly important calciums are in your muscles. They also are really important in your nerves because it turns out it's calcium that causes your nerves to release the neurotransmitters in the first place. Calcium is the secondary messenger that tells the nerves, hey, it's time to give off that neurotransmitter. Calcium is also involved in cell division, mitosis, meiosis. But for right now, what you really need to know is that the main secondary messenger is CAMP, cyclical AMP. But in a few places, like in your muscles and your nerves, calcium can also fill that role. It can be a secondary messenger. All right, last slide we'll look at probably maybe one more. Responses. The whole purpose of a cell signaling pathway is to get a response. You can have several different responses. Cell signaling can, most of the time, causes certain genes to be turned on or off. It may cause, like in adrenaline with the liver, we talked about how it caused the breakdown of something, like the breakdown of glycogen. It may cause certain enzymes to either be made or to be turned on. And what do we know enzymes end up turning on? What do enzymes do? Say again? No, well, not necessarily. Some of them cause things to get broken down, but just as many cause things to get put together. So the point is they start up what? What enzymes start up? Reactions. So another response is to start some kind of reaction, or maybe turn off some kind of reaction. And we'll talk next time about how some of these signals also cause certain ion channels to be opened. What's an ion channel involved in? What are channel proteins involved in? Facilitated diffusion, the movement of certain kinds of things. This turns out to be really important in the nerves, and that's the last cell signaling point we'll talk about. We're not going to get there today, I don't think. But we'll talk about those ion channels on Tuesday. But the key thing, these are all things that a signal can cause to happen in your body. I'm going to stop there.